Okay, so we are actually 2 minutes ahead, but if it doesn't matter, then we will start the lecture series right now. Would that be okay, Pak Triwibowo? All right, then. So, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, dear ladies and gentlemen, and welcome back to the online cast lecture series brought to you by the Tropical Medicine Postgraduate Program, Faculty of Medicine, Public Health and Nursing, Universitas Gadjah, Mada, Indonesia. My name is Rima, and it always been an honor for me to be your MC today. Ladies and gentlemen, we are going to have a talk on the importance of surveillance vectors and vector control for the control and elimination of vector-borne diseases. That will be delivered by Professor Neil F. Lobo. He is a research professor, and today's session will be moderated by Bapak Triwibowo. A brief introduction to you. Pak Triwibowo Ambargarjito, Doctor of Philosophy. He is a researcher at the Vector-Borne and Zoonosis Diseases Research Group, Research Center of Public Health and Nutrition, Health Research Organization Indonesia, or we are more familiar with Badan Riset in Brain. So he earned his undergraduate from Universitas Gadjah Mada, and then Master of Health Science in Immunology and Molecular Biology of Tropical Diseases here at Universitas Gadjah Mada as well, and his Doctor of Philosophy at the University of Montpellier, France in 2022. So his research is most of them is about the vector diseases and as well as the public health. So without further ado, ladies and gentlemen, please welcome Bapak Triwibowo Ambargarjito, Doctor of Philosophy.
Okay, thank you very much, Risma. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, especially for Professor Lobo, and also ladies and gentlemen. So I'm honored to be a moderator for today's lecture featuring Professor Lobo. The topic of his lecture is importance of surveillance vector and vector control for the control and elimination of vector-borne diseases. So we know that vector-borne diseases pose significant threats to global health, and understanding the biology and taxonomy of mosquitoes is crucial for effective control strategies. So we're happy because of Professor Neil Lobo now is together with us. So Professor Neil Lobo is a research professor Biological Science from Department of Biological Science, Eck Institute for Global Health, University of Notre Dame, Indiana, USA. And Professor Lobo is a renowned expert in the field of medical entomology and has made a significant contribution to the study of mosquito and the dynamic transmission of the disease, especially for malaria and some other vector-borne diseases. And his research focused on malaria, especially in an endemic country. Actually, Truk centered primarily on the entomological driver of transmission. His research seeks to integrate entomology with other key aspects of transmission, including intervention presence, epidemiology, and human behavior, and also weather. Through this integration of data types and use the modeling, his research seeks to elucidate how these various vectors of malaria transmission interact, better understand root causes of persistent transmission, and improve protection on highly granular local level. So actually, he has a lot of experience related with the many research regarding the vector-borne diseases. So started with the transgenic and genomics, with studies this, and then focuses on vector species, and then bionomic, vector population biology, and then control and elimination strategies. And of course, human behavior, including large-scale entomological and epidemiological trial examining multiple intervention paradigm, and so on. There's a lot of experience and a lot of publication from the Professor Neil Lobo. So I think this is time for us to hear from his lecture. So let's for me welcome Professor Lobo to the stage without further ado. So Professor Lobo, time is yours.
Thank you very much, Patri.
Okay.
Thank you very much for that introduction. So, hey everyone, I'm Neil, and as Patri said, I work in many places, so I'm traveling a lot. So right now I'm actually in India. But today, just as a clarification, Patri, how long do I have to talk?
Maybe it's more than yeah, I think it's about 2 hours. Yeah.
Oh my gosh.
Yeah, but maybe you can arrange maybe 1 hour or 30 minutes.
Sure.
Okay. Yeah.
So if everyone wants to take a nap in the middle, that's fine. Just kidding. Cool.
Okay.
So I will share my screen. I should be able to share my screen, right?
Okay. Go ahead.
Let me share my screen. Okay, it's taken a second. Share. So today, most of my research and my work is on malaria. So I'll start with talking. My whole presentation will be based on malaria, but honestly, you can apply sort of these principles and the work that I do to any disease to try to understand almost any disease, especially vector-borne diseases, of course. So I'm going to be talking today about the importance of surveillance and vector control when we're looking at the control and elimination of vector-borne diseases. And pay in mind, this is mostly with malaria, but again, applicable to dengue, any vector-borne disease, and other diseases as well. So, okay. And I wanted to start because I really like this saying. Before DDT, malariologists were trained to be problem solvers. But after DDT, malariologists were trained to be solution implementers. And what this means is that the second they got DDT, a good insecticide, people were told, you don't really have to think, just go spray DDT, just go implement this. So people kind of stopped thinking about what the actual problem is and what the actual problem to solve is. And so like for right now, everyone says, just give everyone a bed net, but they don't question how bed nets are not working or how much they are working or what the problems are. So I feel as scientists, we really need to become, as malariologists, we need to become problem solvers again. And to start off, I'm sure you know this, what is the world's most dangerous animal? It's the mosquito. And this is an old figure, so the numbers might have changed a bit, but mosquitoes killed the most human beings on the planet. And the second most dangerous animal, I think this is funny, is humans. The humans kill the second most humans on the planet. So actually, mosquitoes are really, really dangerous to us humans. And just as a starting point, right? The vector interventions. When we try and reduce disease, we have interventions like bed nets and indoor residual spraying and things like that. Why are they so effective? The usual answer is that because it kills them. And this is true, but it's also, when we're looking at malaria, it's actually a little more complicated than that. So and this is a complicated figure, but the quickest version is an infected mosquito on the left bites a human being, and it takes about 12 to 14 days for the malaria inside the human being inside the mosquito to become dangerous to humans. So a mosquito, if it bites me today, it's only infectious to other human beings after about 2 weeks. So the part about why vector interventions are so effective is because of mosquito lineage. So think about it this way. If the survival rate of a mosquito is 80% every day about 80% of mosquitoes survive. And then if Plasmodium, the malaria vector, takes about 2 weeks to mature in the mosquito. So mosquitoes are only infectious to humans after those 2 weeks. So therefore, old mosquitoes are more dangerous because they have to survive those 2 weeks, right? So old mosquitoes are more dangerous. Now let's look at this figure. Let's say the mosquito survival rate is 80%. Now on the left side, we have 1,000 mosquitoes. So every day, only 80% of them will survive. And this is what the curve looks like. The survival rate of those 1,000 mosquitoes over, let's say, 22 days. So now let's imagine on day 2, every single one of those mosquitoes bites me and I have malaria. So all those mosquitoes will have malaria. Now, that malaria takes about 2 weeks to develop in the mosquitoes. So those mosquitoes, all those mosquitoes that have bitten, infected me, can bite everyone, anyone, every single day for those 2 weeks, and no one will get malaria because they're not yet infectious. The plasmodium or the malaria has not yet developed. So after those 2 weeks, you can see only that proportion of mosquitoes are infectious to humans. They can actually infect humans. And that's about only 1 to 2%. And remember I said about all the mosquitoes would bite an infected person. And in reality, that's not true, right? Only a small proportion of them would bite an infected person. So the red proportion is an overestimate of infectiousness. So now let's say we have an intervention that reduces the survival rate from 80% to 15%, just 15% difference. And the green is the 15% survival curve. And you can see that the infectious mosquitoes is not 15% smaller than the 80% survival curve, right? It's a lot smaller. So a small increase in the death rate can have a huge impact on the age structure of mosquitoes. So older mosquitoes are more dangerous. And if you reduce the number of older mosquitoes, you'll have a lot less disease. So interventions against mosquitoes like bed nets and spraying actually reduce the age structure, and you have less infectious mosquitoes. And you can apply this to any disease, actually, based on the incubation period. So you can even increase the survival rate just to reduce the survival rate just to 50% every day. And you can see you can have a lot of mosquitoes, but no malaria, because the mosquitoes don't survive that long. So you can have a lot of mosquitoes and no malaria. I hope that made sense. So that is why vector interventions are so important. And as I'm trying to go through a couple of principles and how I apply this to my research, to my work, to trying to understand disease transmission and reduce disease transmission. And my next question is, why are mosquito behaviors important? And here's just a simple example. So here's a little picture of a village in which you have all people indoors and people outdoors, and you have a cow, and you have all those orange mosquitoes flying everywhere. Now, let's say at this site, there are 3 kinds of mosquitoes. Let's say there's one that feeds outdoors, that's the blue ones, one that feeds indoors and outdoors, the reddish ones, and one that feeds inside only, the yellow ones. And this is actually true because there are different vectors of malaria have different behaviors. So for example, in Kenya, we actually have 3 species of mosquitoes that have these exact behaviors. And in Asia, we have similar mosquitoes with similar behaviors. Now we have to remember that interventions take advantage of susceptible mosquito behaviors. For example, bed nets take advantage of mosquitoes that come inside the house and feed on people when they're underneath the bed net. So late night feeders, right? If a mosquito does not come inside the house, the mosquito will not be impacted. If we have IRS, which is indoor residual spraying, where we spray the inside walls of houses with insecticide, and the mosquito, when it sits on the wall, it will get exposed to insecticide and die. Now, if that mosquito does not sit on the wall, it will not die. If the mosquito does not come inside the house, it will not be impacted by the intervention. So you can see how these different behaviors will allow a mosquito to either be exposed to the intervention, or it can actually cause it to escape the intervention. Now let's look at our little scene over here, where we have 3 kinds of mosquitoes. Now let's say we put in indoor intervention, the bed nets. Now the bed nets are only going to impact those mosquitoes that go inside and am I making sense? I hope I made sense. They'll only impact those mosquitoes that are going inside the house late at night and when people are underneath those bed nets. So it's going to impact those 2 species. And the third one that's feeding outdoors, it really won't impact them that much. So you can see how interventions take advantage of mosquito behaviors, and the overlap between the intervention And the mosquito behaviors are really important and critical to how effective that intervention is going to be. And so interventions only affect vectors with those behaviors, right? The behaviors that make them vulnerable to the insecticide or to the intervention. And here's an example. So this is data from Kenya. And what we see on the left, the black is the proportion of one species, Anopheles gambi. And the green is the proportion of another species, Anopheles arabiensis. And at this site, there are actually 3 species of mosquitoes, just like in the figure I showed you. Anopheles gambi loves biting humans, also cows, and it feeds inside the house a lot and outside as well. Anopheles arabiensis feeds outside mostly and likes cows, but it also feeds on humans. So you can see on the left side, you can see the proportion of Anopheles arabiensis down and the proportion of Anopheles gambi above. And you can see most of the population was Anopheles gambi. And then they put in bed nets, Ella lions in our bed nets in about in 1997, 90, in the middle of 1996. And you can see immediately the proportion of species started changing because Anopheles gambi that feeds indoors and likes feeding on humans was being really impacted by the intervention, by the bed nets. And suddenly the proportion of Anopheles arabiensis went up. The numbers of arabiensis didn't go up, but relative proportions went up. And you can imagine how that Anopheles arabiensis is now continuing transmission outdoors and avoiding the intervention. What's interesting is Anopheles fenestis, which is not in this figure, loves feeding on humans, really likes feeding on humans, and primarily feeds indoors. And Anopheles fenestis disappeared after putting in bed nets. So Anopheles fenestis was affected much more than gambi and arabiensis with bed nets. So that's an example of how mosquito behaviors are so important. So understanding your species at your site and what they're doing and how they're interacting with humans and your interventions is really important and critical to intervention efficacy. So why are species compositions important, right? And I'm sure you would have guessed this by now, just from the previous example. Here is a figure of the primary vector species for malaria across the world. And you can see, even though in the US, on the left, you have Quadria maculatus, there's lots of vectors all over the US, including right there where I live, by the Great Lakes, but there's no malaria. So you can have a lot of mosquitoes, a lot of good vectors, but no malaria. What this figure not see is all the different species at each site. For example, I'm sure you know, in Indonesia, right in the center over here, it says Sandaicus SL, but there are a lot of other vectors present there. And each of those vectors can contribute to transmission differently. And why are these important? Because different mosquito species have different behaviors. And I sort of just explained this in the figure below, in that little cartoon in the village with 3 species. And here's an example. In the Kenya highlands, they imagined that there were only 3 species of vectors. And by the way, don't pay attention to all the detail in this figure. It's actually, don't worry about the detail. The important part is that people thought there were 3 species. The 3 species I talked about earlier, the one that feeds indoors, outdoors, and one that feeds indoors and outdoors. But what we did when we actually looked at the species composition, we found 17 species. People don't look to see what their vectors are. And we were completely surprised that all the red ones are actually unknown species. We don't even know what they are. We had to call them species A, species K. And the interesting part is species A, the first one right on top, was the one we caught most of. It was the primary vector, and we don't even know what species it is. And if we don't know what species it is, we don't know what vector, what behaviors it has, and we don't know how the interventions are impacting it. So just this basal knowledge of species compositions at our sites and what their behaviors are can be really important in a disease intervention strategy. Yeah, species A was the unknown one and it was the primary vector. So we can have species-specific behaviors, of course. So just a quick summary point on this is that malaria transmission is determined by local vector species, obviously, correct? And different species have different behaviors. And species-specific behaviors and compositions, so the different types of species at your site and the different proportions of each species at your site will determine, this is another point that I'm not talking about, but I will get into it in a second. So species-specific behaviors and compositions will determine where and when malaria transmission happens, as well as how good your intervention strategy is. So as the baseline, we need to figure out our species compositions and their behaviors. And I also talked over here, the little gray point is sampling methods. So when we're trying to find out what species compositions are at our site and what their behaviors are, your sampling device is just like an intervention. It also takes advantage of mosquito behaviors, right? Because if they don't overlap, you can't catch that mosquito. So different mosquitoes have different behaviors. And so your sampling method has to be chosen carefully because you might not catch a bunch of mosquitoes. So that's important to understand as well. So species-specific behaviors and compositions are so important. So here's a question. Besides mosquito behaviors, what is a huge predictor of intervention success or failure, right? People focus so much on the mosquito. And I like to use this picture because the answers in the picture is that here's a bed net and here's a child. This is a picture from Kenya, is sleeping and there's a bed net hanging up, but the child is sleeping outside the bed net. So even though we have a good intervention that can protect the child, the huge predictor of intervention success or failure is human beings. Like, how are human beings functioning with disease transmission? How are they functioning with the mosquitoes? Where and when are they actually active? Are they using the so human behaviors are a huge are very important, and understanding these is really important. And here is an example of what we do in malaria. When we're trying to understand mosquito behavior, what we do is we try and catch these mosquitoes to see what they're doing. And the common way of catching them is called a human landing catch. And it sounds a bit crazy, but what we do is, for example, we're trying to understand which mosquitoes come inside the house and which mosquitoes come outside the house. Like how many mosquitoes bite a person inside the house and how many of them bite outside the house. So we'll actually sit, that we have a person inside the house and a person outside the house, and they'll wait for mosquitoes to come and land on their leg, and then we catch them. And so let's say the person inside the house catches let's say over the night, the person inside the house catches 100 mosquitoes, then the biting rate is 100 mosquitoes per person per night inside. And let's say the mosquitoes outside, the person outside the house catches 20 mosquitoes over the course of the night, then we say the outdoor biting rate is 20 mosquitoes per person per night. But what's interesting is we've been doing this for so long, and we've been doing it in a weird way. We've been doing it wrong. And this is how I'll try and explain it to you. So the human landing catch, let's say between 1 and 2 in the morning, let's say I'm sitting outside trying to catch mosquitoes between 1 and 2 in the morning. And let's say Pak Tree is sitting inside trying to catch mosquitoes. And Pak Tree catches 50%, let's say he catches 50 mosquitoes inside, and I catch 50 mosquitoes outside. So he catches 50% and I catch 50%. So we'll normally say the risk of being bitten inside the house is 50%, and the risk of being bitten outside the house is 50% by these mosquitoes. And we've been doing this for a long, long time. And now I'm trying to change this because I think this is completely wrong. And I'll try and explain why. So in real life, right? Remember, if Pak Tree and me are doing these mosquito catches, it's very artificial. You know, we're sitting there and trying to catch the mosquitoes. But in real life, there are mosquitoes inside and they're outside, and people might be asleep, people might be awake, people might be doing anything. And remember, I said between 1 and 2 a.m., right? At 1 and 2 a.m., where is everyone? Everyone is probably indoors asleep, right? And here's the mosquito catchers, Pakri is trying to catch them inside, and I'm trying to catch them outside. But the community is actually indoors asleep, and maybe they're even under bed nets. So in a weird way, the number of mosquitoes, even if Pakri catches 50% indoors, and I catch 50% outdoors, because everyone is actually indoors asleep, the risk of being bitten is 100% indoors and 0% outdoors, because there's no one in the community outdoors, right? So normal human landing catches, which is what we've done for a long, long time to understand exposure and understand risk of getting malaria or being bitten by mosquitoes. So this is not actually indicative. This does not actually tell us how much the local community is being bitten and where they're being bitten, because their behaviors are so important. So the next point is, as I just mentioned earlier, human behaviors are really important to understand, and how we measure this is also important. So human behaviors are so important because it allows us to identify where and when humans are interacting with mosquitoes, right? Because that is where they are getting bitten. So we can identify these human vector exposure points and periods. And based on that, we can design strategies, right? Appropriate and effective intervention strategies. One thing we can do is, based on human activities, we can have social behavior change and communication strategies where we inform the local people about where they're being exposed to malaria, how they can change their behaviors, better bed net use, and things like that. It also, if we understand human behavior, vector behavior, and the intervention, we can understand gaps in protection. So here is something. So everyone usually says, like, think about this with any disease, right? We have, using malaria as an example, people will say, we have 80% bed net coverage, 90% bed net usage. We're doing such a great job. And I completely agree. You're probably doing a really good job. But my main question is, so if you're doing all this, why do you still have so much malaria, right? Why do you still have malaria if we're doing all this good work? And the answer is because of gaps in protection. Like, where are bed nets not working? Where are humans coming in contact with the vector, with the mosquito, when there's no intervention there, right? So that's a gap in protection. And understanding our gaps in protection from the perspective of human behavior, mosquito behavior, and how the intervention functions is really important, because that'll tell us how to optimize our present interventions, how to get the most protection out of them. And it also tells us what the gaps in protection are, so we can try and put another intervention there. Hopefully we'll have the funds to do that, but that'll tell us how to put another intervention in that space and time. And a simple human behaviors that I measure in a lot of our studies, with respect to malaria, of course, what time people go to sleep, what time they wake up, right? How long they spend indoors and outdoors, if they're actually using the intervention. Sometimes people have changes in behavior. For example, they may have, if during prayer services or religious things, they might be outside all night long and exposed to mosquito bites. Sometimes in the wet season, people are out fishing maybe, or they're out in the fields and they have different exposure profiles. Maybe they have cattle herders and they're moving with their cattle long distances and they're more exposed because you cannot have a bed net in those spaces. So all these human behaviors are things that we try and measure and understand, to understand what are the humans doing? How are they being exposed to the disease? And how can we put an appropriate intervention in that space and time? So what we do is we try and integrate vector and human behavior data to understand both gaps in protection and how people are being protected. So as just to repeat, right, vector control interventions target specific vector behaviors. So let's say this blue oval is an intervention and it overlaps with these vector behaviors. So the intervention will only work in the space where the vectors and the intervention overlap, correct? If you imagine a bed net, It'll only interact with the mosquito when the mosquito goes indoors and if there's a human underneath. So that is where humans come in, right? So when we overlap the intervention, the human and the vector, and what's happening locally, we can see where they're being protected, because that's where, right in the middle, is where protection happens. And we can also understand gaps in protection, because this is where our intervention does not function, or maybe the human's not using the intervention. And this is where we have disease transmission. So understanding this gap in protection is, in a very weird way, even more important to understand what a good job we're doing, right? Because in these gaps of protection is where we get continued malaria. In these gaps of protection is where we have people dying. In these gaps of protection is where our job actually is. You know, this is the problem we've got to solve. This is what we've got to understand, what is happening at our sites. And it might be different in Salatiga, it might be different. It is different in Salatiga than it is in Pakistan. And it's completely different in Brazil. So in every area, both these interventions, the vectors and the human behaviors are different. And so understanding this is super important to having effective strategies. So you can see understanding these gaps in protections is important. And the next step is, let's say we have bed nets rolled out and we still have gaps in protection. Now, what we can do is, when we understand where and when humans and vectors are overlapping, and we have continued malaria or any disease, we can now actually place another intervention there and increase protection there. So the second oval below is another intervention. So in this sort of way of thinking about this from a problem point of view, trying to understand what's happening locally, we can understand where all our malaria comes from, how to optimize our present interventions, as well as layer interventions, put in multiple interventions at a specific time and space so that we can actually protect more people. I hope that made sense. And I'm going to show you an example of what we did in Cambodia. So in Cambodia, it's a low transmission area country. They're aiming to going towards elimination. And most of the transmission actually happens in these forested sites, where people go to the forest, where they do logging, they mushroom hunting, all these forest activities. A lot of it is illegal, so it's kind of hard to work there. But a lot of it is, that's where most of the malaria transmission happens. And you can see they live in these very temporary structures. And so it's really hard to have a bed net there. And it's impossible to spray it for insecticides, and it's so open. So it's really hard to work with. So we wanted to understand what kind of interventions could we possibly use here? What kind of interventions could we try and, implement here that's applicable to this population, applicable to the mosquitoes there? So we went there and we looked at the behavior. And what we saw is that we could compartmentalize it into three parts. One, where they're sleeping under these contemporary structures. Two, when they're outside these structures, where they have cooking and they eat and they drink and they kind of just hang out, where they relax outside these structures. And then three, when they went to the forest to do their work, to log, to hunt, or to look for mushrooms and things like that. So now our job was to try and figure out what interventions might work in these spaces and times based on human behavior. So in these temporary structures, we thought of a spatial repellent, a VPS, which is a volatile pyrethroid spatial repellent. We had a spatial repellent spray, and we had insecticide-treated clothing, as well as a topical repellent. A topical repellent is something you put in your skin, like Autan. I think in Indonesia it's called Autan. So in these structures, these interventions would work there. When they're outside these structures, some of them would still work, like the passive volatile pyrethroid spatial repellent, the spatial repellent spray, like treated clothing, as well as a topical repellent. Now, as a note, the passive VPSR is a spatial repellent that you just hang up and you leave there. And it can last a month, 2 months, 3 months, depending on the product. So the good thing is, you don't have to rely on human behavior to use it every night like a bed net. You can just leave it there. So that a passive product that doesn't require daily action from the human point of view is actually more effective, because a lot of the times a human might forget or they don't want to use things. For example, the spatial repellent spray, you actually have to go spray it. And if you imagine that you don't have to spray it and it's always working, the efficacy would be higher because there's more compliance, there's more usage. So outside these temporary structures is what these four might work. And when we went to the forest, the treated clothing and the topical repellent would work. And we did trial them in a semi-field system in a sort of like a big insectary. And we found that these actually did work. And now the next step would try and have a package of interventions and trial them in the field. So this is in Cambodia in the forest. And you can see it's clear cut here. And each of these structures, the white tents, had a person with a single intervention in there. And one of these structures had all together as a package. So we wanted to see the number of mosquitoes that would approach each one of these, the person in each one of these tents. So we can see how effective this is. And we compared it to a control. And the control person had nothing, like they didn't have any intervention. And this is a Latin square design. So every night we'd move the intervention, one structure, one structure, one structure. And as a note, the black one in the middle, you can see that small black structure in the middle is where I slept, where we all slept for, I think it was more than 4 months while we did this study. And you can see it's really open and the wind can blow through and it's very temporary and very sort of not a house. And if we can protect people in these spaces, we can really protect people well in houses. And here is baseline data. So you can see the x-axis is the number of days, 49 days. And the y-axis is the cumulative number of anopheles per night. So every day we accumulated the number of anopheles. The yellow line is the control, which is 3,537 mosquitoes. We caught 3,537 anopheles, which is a lot. These green and blue lines are different other interventions alone. The treated clothing, the spatial repellent spray, right? All those other interventions. And all of them had a really good impact. But if you remember, each of them functions in a certain way and does not function in a certain way. What was interesting is when we put all together, it worked really well because it covered multiple spaces and times, right? And multiple human behaviors. So the combination caught only 124 mosquitoes compared to 3,537. So we had a reduction of almost 95%, which is really good because we used data. We went to see what the humans were doing. We tried to see what the vectors were doing. We tried to pair the interventions to the humans and the vector behaviors. And you can see the efficacy we got, which was actually really good. So right now, what's happening is we're taking this package of interventions to the field and actually implementing it in communities in Cambodia, and hopefully it will be useful. Yeah, so here's the big question, right? How can vector surveillance be used as an intervention? Can we actually use surveillance as an intervention? Instead of, bed nets are an intervention, IRS is an intervention. Can we use surveillance itself as an intervention? So the reasons why this is good and useful is because when we conduct surveillance, we actually are collecting data, right, in real time. So we know what's happening at the site right then. So if we collect data and we know what's happening at that point in time, we can make evidence-based decisions. And based on that data, we can actually target and tailor our reactions, our strategies to the disease transmission that's happening. So targeting and tailoring, you might hear that a lot from WHO nowadays. How do we target and tailor our approaches to disease transmission? For example, WHO says to give everyone a bed net. But as we just discussed, bed nets only work in some places and at some times, right? So what do we do in populations that don't want bed nets? We have to target and tailor our responses, try and figure out what they will actually use, things like that. So we'd also understand through vector surveillance how our present interventions are functioning. Are they functioning well? Are they not functioning well? What are the gaps in protection? And for example, with bed nets, if people have a bed net and don't use them, then we can use SBCC, or social behavior and change communication, to try and optimize their usage, try and increase their usage. So now we can use surveillance to optimize our present strategy, to increase protections. And this way, we don't really waste too much money, right? If we're already spending a lot of funds and capacity on distributing bed nets, on spraying structures, on whatever intervention we're doing, we want to make sure we're getting as much out of it, as much protections out of it as possible. So surveillance can actually help us optimize our present strategy. One thing that we almost always don't realize, and it took me actually a minute to actually realize, is that when we do conduct an intervention, when we do something to prevent disease, we actually change transmission, right? And if we change transmission, why do we continue doing what we have done before? Why do we do the same thing after we change transmission? It makes sense that we adapt what we are doing to changing transmission. Because if we do the same thing again and again, it's not going to actually impact anything more. So it changes transmission in 3 specific ways. It impacts, at least with malaria and vector interventions, it impacts vector compositions. And I think this is the example I had where bed nets or LLIs impacted the vector species compositions, right? The ones that were impacted most had susceptible behaviors. That was Anopheles gambiae. And the ones that were feeding outdoors and were outdoors and did not go indoors, Anopheles arabiensis, were not impacted by the intervention. So their species compositions changed. There were more of the ones that bid outdoors and less of the ones that bid indoors. And it was reversed, right? Sorry, I said that wrong. What I meant is there were more that bid indoors first and less outdoors. And after bed nets, proportionally, the outdoor ones went up. My mistake, I made a mistake there. So now with vector behaviors are also something we can change. And what's interesting is that, just imagine, if we have mosquitoes that bite indoors and outdoors, and we put an indoor intervention. Now we're killing off all the indoor feeding mosquitoes. Now that population is going to change its behavior to outdoor feeding more because we're killing off all the ones that feed indoors, right? So all the ones that feed outdoors more are going to survive better and there are going to be more of them. And this is actually exactly what we see at some sites. So this little figure is from Solomon Islands in the Pacific. And the x-axis is the time of the night. So it's 6:30 p.m. to 6:30 a.m. and 24 is midnight. And the number of mosquitoes biting per person per night is on the y-axis. So you can actually see over the course of a night, most of the mosquitoes by the red star were biting at midnight. So most of them bit at midnight. So now the Solomon Islands government implemented bed nets and indoor residual spraying to kill mosquitoes. Now, all of a sudden, from 1971 to 1972, right, all the mosquitoes started feeding outdoors. Started feeding early in the evening, not at midnight. And they feed early in the evening, and I'm sure you can imagine why. Because at midnight, everyone is asleep and under their bed nets, and the mosquitoes try to bite them, and they die. But early in the evening, people are not asleep. Let's say this is 6:30, so maybe 7 o'clock. People are not asleep. They're outdoors. They can be bitten by mosquitoes. They're indoors and they're awake. They're doing things. They're cooking, they're eating, they're playing. So they're not under their bed net. So this is where mosquitoes can actually get their food. So all the mosquitoes that were feeding in the middle of the night, their population went down. And we changed that mosquito population behavior to early evening feeders. And actually, it's not just early evening. We change them from indoor midnight to outdoor early evening. And I haven't demonstrated that data on this picture, but trust me, the mosquitoes change their behavior from feeding in the middle of the night, indoors, to outdoors and early in the evening. So vector behaviors can change. And you can imagine how you'd want to survey and monitor your vector behaviors, because that is where your vector interventions will stop working, when the mosquitoes change their behaviors. And the third one is what we measure a lot, insecticide resistance. So we put an insecticide in the environment, all the mosquitoes that are susceptible will die, and all the ones that actually are resistant will survive, and their population will grow. So just like the behaviors, we are technically selecting for species that can survive our interventions. We are selecting for behaviors that can avoid the intervention, and we're selecting for insecticide resistance so that they can survive even if they're exposed to the intervention. So when we implement an intervention, we are basically changing transmission. And this is evolution happening, right? Evolution is pushing back. It is changing the compositions and behaviors and insecticide resistance. So we have to battle evolution here. And we have to understand all the changes that are happening as soon as we can, so we can change our responses, because we've changed transmission. Yeah, I hope that made sense. Yeah. So now when we do surveillance, we can actually understand responses. Once we see how the transmission system is changing, we can change our responses so we are more effective, so that we can protect more lives. And at the same time, we can look at the potential of other interventions, right? So now in this little scenario over here on the right in the Solomon Islands, where initially bed nets would be very effective indoors late at night, now the mosquitoes are biting outdoors in the evening, we have to try and figure out what intervention might work in that space and time. So that is the potential of other interventions. We try and figure out what will work there. So here's a quick figure of all the different interventions that are sort of in process right now, and I'll go through them quickly. So in the center, you have a blood-fed mosquito, and the two WHO approved interventions right now are bed nets or LLINs. Those are the ones in orange, and IRS, which is indoor residual spray. Even though I know this is not an IRS spraying device, it's the only little cartoon I could find. So bed nets and indoor residual spraying, along with larval source management as a supplementary intervention, are the main WHO recommended interventions with a focus on bed nets. So WHO right now, there are a couple of other interventions that are in the pathway to being approved, and I'll go by them one by one. Transgenic mosquitoes, or genetically modified mosquitoes, are one avenue where we can look at producing a vector that is either not capable of transmitting the disease, or we can try and kill them off through genetic manipulation methods. For example, we can release sterile males, or we can release mosquitoes that mate with females and kill the females, or all the eggs will not survive. So transgenic mosquitoes, this is actually quite far from being approved because all the trials are not done yet. And this might actually only be ready in 15, 20 years if it becomes ready. Now, attractive toxic sugar baits are another intervention that are in trial right now. I think the trial data from Kenya is going to be today. It might be today that they're actually yeah, no, it might be this week, I think. I'm not sure exactly where they're actually looking at the data this week. So what's an attractive toxic sugar bait? So mosquitoes feed on blood. Female mosquitoes feed on blood because they need protein to lay eggs. But all mosquitoes, male and female, feed on sugar, feed on plant juices. And this is where they get a lot of their nutrition, their carbohydrates from. So they will feed on plant nutrition, plant sap. You might see them sitting on flowers and they'll be actually sucking out the nectar, just like butterflies, we'd imagine that. So attractive toxic sugar baits is basically a device that has something sweet in it, a sweet, but it also has an insecticide that will kill the mosquito. So all these mosquitoes that will be attracted to this to feed on the sugar will die. So at present, there are trials going on in Kenya, in Mali, to try and see if this can actually reduce disease. The trials are in process. Endectocides. So endectocides, like an example of an endectocide is ivermectin. It's actually a drug. And this is used for helminth infections, for nematode infections. So this kills worms. And a lot of times people will treat their cattle with it. They'll treat their sheep with it, and to kill off all the worms in the cattle. But what this also does is any mosquito that feeds on cattle that have been treated with an endectocides, the mosquito will die. So we can actually use endectocides to kill mosquitoes by treating cattle. So right now, there are trials going on in Mozambique, in Kenya, and in somewhere in Asia as well, where they're looking at how treating either cattle or humans, because humans can take insecticides as well. So if I take an insecticide, any mosquito that feeds on me will die. So the only problem with insecticides is that it has a really short half-life. So it only lasts a couple of days in my system. So they're making more longer lasting formulations now. So for example, if I take the medication, I'll be able to kill mosquitoes, any mosquito that feeds on me, for a much longer time instead of just a few days. I'm gonna have a quick sip of water. So endectocide trials are still in process. And I think a couple of them are almost done or already done. Spatial repellents. And you all might know what these are. A spatial repellent is an insecticide-based intervention that volatilizes, right? So it volatilizes and goes into the air, and you sort of have like a bubble of protection around you. And since it's toxic, and since it might smell really bad to the mosquitoes don't want to enter the space. And so the biting rate, let's say if I'm in this room here and I put it in a spatial repellent, the mosquitoes don't want to enter. So it repels the mosquitoes. It can also possibly kill them or disarm them, because if they're exposed to something that's a poison or toxic, they may not feel well. So they may not feed for another day. They may go hide somewhere because they just need to recover. They may also die. So spatial repellents have protective spaces around them. And people inside those spaces have less mosquito bites. So right now, the spatial repellent trials are going on in Mali, in Kenya, Uganda, and in Sri Lanka. We finished the trials in Peru and one in Indonesia a couple of years ago, where there was so little malaria in Indonesia. We actually had an effect, but it wasn't statistically significant. And the one in Peru against dengue actually showed a statistically significant impact, which was quite good. So spatial repellents do work, and they're expecting WHO approval in 2025, I think, after the trial results come out, of course. So these are the interventions that are approved by WHO, and the others are now coming out, though the one that might take the longest is transgenic mosquitoes right now. Yeah, and I thought that I could give you an example of how entomological surveillance can actually be really effective. And I hope I don't finish too soon, because I don't think I prepared to talk for 2 hours, but I'm happy to have a long question and answer session. Yeah, so here's the example from Panama. So I'll just go over briefly in what is happening in Panama. So Panama actually has low malaria, and in 2001 to 2004, there were just malaria outbreaks. There are about, let's say, every year from around 900 to 5,000 cases, depending on the year and where this was happening. They had a strong anti-malaria campaign, and by 2016, cases went down a lot. there are only 769 cases and no deaths. So that is actually really good. However, in 2019, cases went up about double to 2,000, and all this malaria was in a few specific areas, not across the country. And these areas were rural and remote areas, and the people who lived there were indigenous people, the tribal people. And these tribal people are strong characters, and they're really lovely people. And they are people you can't just convince them to do anything. So they did not want to use bed nets so much, and they lived in the forests, and so they were very exposed to malaria. And 90% of the malaria cases in Panama are in these populations. And the picture on the right is this lady who had, they wear these really lovely beaded, all these are beads, and they're not pants, but they're like just on their legs and arms, they have these beaded things. They're quite nice. They're lovely people with the most malaria in the country. So this is the malaria case outbreaks where they were across Panama. And the purple you can see is where there were outbreaks. And as you can see, all the cases were in these forested areas, and all the cases were in these indigenous tribal communities. The bigger the circle, the more number of cases. And we did our study over here where the red arrow is, right by the border with Colombia. Another sip of water. Am I speaking too fast? Am I speaking too slow? Am I okay? Does everyone
I think
understand?
it's okay. Yeah, it's
Okay,
very good. Yeah. It's
okay, cool.
Yep.
Cool, thank you. Okay, just making sure. So we conducted our study, our entomological surveillance at this site here. So this is what it looks like. These are what the structures look like where the community lives. And so they live close to the sea. Sometimes the houses go into the water, and then the other side would be forest, which they'd go into the forest to do all their hunting, food gathering, and etc. But they're very exposed to malaria. So I think this is the wrong slide. Here. So here's a picture of these three ladies in the village. So I was there for many months, and every day, here's a funny story, they would sit there and just stare at me. You can see the beads on their legs and arms, and they just stare at me, and they'd never talk to me. And every day, these three ladies would pull up these chairs, and while I was working there, counting mosquitoes and everything, they just sit there and look at me. And then at the end, before I was leaving, I went up to them and I was like, excuse me, do you mind if I take a picture of you all? Because you look so nice sitting here every day. And for the first time in all the time I was there, they talked to me. And guess what they said? I said, can I take a picture? And they said, $10. It was quite funny. They were probably posing there for months and months, hoping that I would take a picture. But look at that. It's a nice picture. $10 picture. They were really nice. So in Panama, let's look and see what's happening from the vector side, from the from the intervention side of things. Bed nets, ITNs, or insecticide-treated nets, they're available in the market, and people buy them, but the Ministry of Health does not supply bed nets to the community. So if there are any bed nets in households, the community is buying it themselves. So the main intervention that the government does is indoor residual spraying that I talked about earlier on. Now, indoor residual spraying, you can see the picture of the person on the right. He has a big can, and inside there would be a chemical, depending on the country. In Panama, it is phenethrothione, which is a pyrethroid chemical, insecticide, and you spray the insides walls of the house with this insecticide. And what happens is that any mosquito that comes into the house, if they land on the wall and sit on the wall, they get exposed to insecticide and they die. So that's how it works. IRS is the primary intervention strategy implemented in Panama for over 25 years. They've been using phenethrothione the entire time long, and they are reporting no insecticide resistance after 25 years, which was very interesting to me because you'd expect insecticide resistance after that amount of time. They also do larviciding, but it's not really done in a big way, and they just do it here and there. So they don't really know how effective it is. They don't really know how effective bed nets are because they don't implement them. They believe IRS is effective because there's no insecticide resistance, but malaria was actually going up, right? And it doubled in the year before we did the study. So the Ministry of Health came to us and said, so we need to understand why our malaria is going up. We actually got it to go down, but now it's going up and we need to understand it. So what we decided is to see, first of all, ask the Ministry of Health, what is your question you are trying to answer? And the question they were trying to answer are bed nets an effective vector control tool. So they wanted to know if they should start distributing bed nets because malaria was not going down. The IRS they were doing, you can only do IRS, they could only do IRS once a year. And the second they had outbreaks, they did not know what to do. They could not go and redo IRS. So they wanted to know if bed nets would be an effective vector control tool. So what we did is we took this question and we are trying to solve this problem, right? So we want to understand how bed nets function, how the mosquitoes are interacting with the bed nets, and the third part is how the humans are functioning there. So what are mosquitoes doing? What are humans doing? And what are bed nets doing in this transmission system? So we did what on the left side is how things are working, and on the right side, how things are not working. So on the left side, bed nets or LLINs stand for long-lasting insecticide-treated nets. So bed nets work, they kill late night indoor biting mosquitoes, correct? So if we understand this, we can look at the proportion of mosquitoes that bed nets are killing. And how do bed nets not work? Bed nets do not work on early evening or outdoor biting mosquitoes. And this is a gap in protection. And early evening, meaning because people are awake, they're outside, and people are not under their bed nets. Correct, I hope that made sense. So how do humans function with bed nets? So humans have to be indoors and under the bed net for the bed net to be effective. If people are not indoors, or if they're not under the bed net, basically, if people are not on the bed net's not effective. So we can look at the proportion of people who are actually using a bed net. How long do they use a bed net? When do they use a bed net? When do they go under a bed net? And we can understand how bed nets are functioning in that community. And the opposite is, of course, if humans are not under the bed net, they're not protected. That's a gap in protection. So we want to understand that as well. And the third part is the mosquitoes. So mosquitoes have to be susceptible, right, to the insecticide. If they're resistant, it's not going to be effective. So susceptible mosquitoes will be killed and resistant mosquitoes will not be killed. And so again, we want to understand both how things work and how they don't work. And yeah, and this is what we did. So we just sort of broke this down like a problem. And again, we can do this with anything, with any question. So the three main questions are, what are the mosquito behaviors? What are the human behaviors? And how are the mosquitoes resistant or susceptible to the insecticide on the bed nets? So what we did is, so here's a question. We have three questions. Now, what indicators, what things do we have to measure that will answer our question, right? What are our indicators? So to understand mosquito behaviors, we want to understand what time they're biting and when they're biting. Right. Inside, so what time they're biting, or inside and where they're biting. I'm sorry, I made a mistake there. So we want to understand if they're biting inside the house, outside the house, and what time. So the human biting rate inside and the human biting rate outside, and the biting time. And if you remember, we use the HLC, which is the human landing catch. If I told you earlier on how we sit outside and let the mosquitoes bite us, to understand that. And we also did human behavior observations, right? And that is, if you look at the indicators, we're looking at where humans are by time. At 6 to 7 p.m., how many people are indoors? At 6 to 7 p.m., how many people are outdoors? From 7 to 8 p.m., how many people are indoors? How many people are outdoors? From 9 to 10 p.m., are people using their bed nets? What time do they go to sleep? Are they not using their bed nets? So for every hour of the night, we looked at the human biting rate inside and outside houses. And for every hour of the night, we calculated and looked at the number of people in each of these behaviors, whether they were awake, where they were asleep, where they're indoors, where they were outdoors. And so our sampling methods, so those are the indicators we collected. The sampling methods are what methods we would use to collect the indicators. And our sampling method to catch the mosquitoes were human landing catches. And to understand the humans was human behavior observations, HBOs. And we did these indoors and outdoors. And through these two methods, we can look at the human and mosquito behaviors. So the next step is the sampling design. So we did 3 time points, 1 in the dry season and 2 in the wet season in a year. And because the funds for the study was very small, we only had $50,000, you can see all the way at the bottom. We did 3 time points. We did 2 houses at each site and 5 days each collection per time point. So we went to each of these sites. There were site selection. If you look at site selection, there are 2 key sampling sites with separate cultural inhabitants. And I'll talk about that in a second. So what we did was we went to each sampling site 3 times a year. And at each sampling site, we went to 2 houses. And we did these collections, the HLC and HBO collections, for 5 days at each time. I hope that was clear. And why separate cultural inhabitants? So in Panama, at these sites with the most malaria, it's a very tribal site where people are very tribal. And there are 2 kinds of sort of groups of people there. One of them is the ethnic tribal, so the ones that live in the forest. And the other ones are descendants of slaves that were taken there, who also live in the forest and in and around these communities. And they're very rural as well. So there are people who are mixed with the local people, but they form a separate group. They have different villages. So there were 2 key separate cultures in these areas. We wanted to sample both because they might have different behaviors. So with capacity and funding, we got $50,000 and we decided that this is a good study. We have questions, we know how to collect, we know what data to collect, we know how to collect it with our sampling methods. We have a sampling design to appropriately collect all the data representing seasonality. We have our sites and we have our very limited funds, but we had funds, which is important. And I'll go over the data that we got so far, right after I have a sip of water. So the key findings. So this is just the vector behaviors. So we had 2 sites. Site 1 is on the left and site 2 is on the right. So if we look at these 2 graphs, from 1 to 13 at the bottom, 1 is 6 p.m. Actually, it's 5:30 p.m. and 13 is 6 a.m. So this is over the course of the night, right? The number of mosquitoes, anopheles per person per hour. So the number of anopheles we caught per person per hour is graphed out over the whole night. The green line is outdoors, the red line is indoors. So you can easily see at site 1, vectors are biting indoors and outdoors. Most of the biting is happening outdoors, and most of the biting is happening early in the evening. What was interesting is that site 1 is actually really close to site 2. So the mosquito behaviors were actually the same. Most of the vectors were biting both indoors and outdoors. The primary biting is outdoors, and the primary biting is in the evening. Site 2 had a lower number of mosquitoes because there were less larval sites there, but the behaviors were basically the same. Now let's look at the human behaviors. Now this is a little complicated, at least it looks a little complicated, but I'll go through it. So again, the x-axis 1 to 13 is the same number of hours that we looked at the mosquito behavior. And the left side is the proportion of people in each of these behaviors. So we look at the blue line. Let's start with the green line. We look at the green line. This is the proportion of people outdoors. So at hour 1, at 5:30, you can see that almost all, about 0.9, about 90% of people are outdoors. And then over the course of the night, that number goes down because they're all going indoors. And you can see they're waking up at about hour 5. They're at 5 in the morning, they're sort of waking up and that starts going up. So you can see the outdoor behaviors of these people by hour of the night. Now the blue line, this one here, is people that are indoors awake. So at the beginning of the evening, you can see there's less people indoors, and then they go indoors, and that number goes down because now it's not that they have gone away, they're still indoors, but now they're going to sleep, so that number goes down. The blue line is people indoors awake. Now the red line and the purple line are people asleep. You can see they all go indoors and they're going to sleep. The difference is that the red line are people not under bed nets, and the purple line are people under bed nets. And just from this simple behavior observation, you can actually see when people are outdoors, when they're indoors, when they're awake, when they're asleep, and if they're using interventions. If you remember, the question was, will bed nets be an effective strategy against mosquitoes here? So these are people who went out and bought the bed nets themselves. These were not supplied by the Ministry of Health. And just from this simple figure, you can see a lot of people do not use bed nets at site 1. Now let's see the same thing for site 2. You'll notice a slight difference. Oh, I'm sorry, I forgot about something. So now if we take this green line, right, and we add up the number of people at each hour, we can actually make a pie chart, right, with the number of people inside awake for each hour, the number of people asleep with and without bed nets. We can have a pie chart and we can look at the proportion of time people spend in each of these behavior groups. And that's what it is. So for site 1, you can see the red is people asleep without bed nets. So most of the night, people are asleep without bed nets. The purple line is people, this line here, is people asleep with bed nets. You can see this is the proportion of people protected by bed nets over the course of the night. The green is people outdoors, and the blue is people indoors awake. So just from this, just from human behaviors, without any mosquito behaviors, without intervention data, you can see that most of the time, people spend their time asleep without bed nets. And about slightly, maybe 23% of the time, they're actually protected. So people are only protected about 23% of the time over the course of the entire night, which was really interesting because no one had collected this data before. So now let's look at site 2. It's really interesting, at least to me. So site 2 looks slightly different, even though it's the same behaviors. You can see instead of 90% of the people outdoors, it's about half the people are outdoors. And you can see them all going indoors. And here, the blue line is people awake. The red line is people with asleep without bed nets. And the purple line is people asleep with bed nets. So you can, if you remember, the figures looks a little different. And this different is really important, actually, because it's 2 communities, the same mosquitoes, but different behaviors. You can immediately see the primary differences is that site 1 went to sleep early and site 2 went to sleep slightly later, right? And the small difference had a huge impact. Also, site 1 used less bed nets. You can see the red line is much higher. And site 2 used more bed nets, about 50%. And site 1 was about 70%. Site 1, 70% did not use bed nets, and site 2, about 50% did not use bed nets. How did this result? How did this impact exposure to mosquito bites? So now, this is just for site 1. We take the human biting rate on the left, if you remember that figure, where green is outdoor biting and red is indoor biting. And we take the human behavior, the figure we just went over, and we do an analysis where we combine the human behaviors in each of these behavior groups with the mosquitoes that are biting in that location for that hour, and this is what we get. So the red line is the biting rate indoors when people are asleep without bed nets. The blue line is the biting rate indoors when people are awake. The green line is biting rate outdoors, just outdoors, biting rate outdoors. So immediately you can see the red and the blue, that's biting indoors. So even though if you look up at the mosquito behavior, the biting rate, the green line, even though much more mosquitoes are biting outdoors, the main exposure is happening indoors when people are awake and when people are asleep, not using bed nets. Right? And so this is a change in thinking, how we think about exposure, how we can combine mosquito and human behavior. This is what we're trying to implement and understand. So the next slide is for site 2. Do you remember site 2 went to sleep slightly later? Correct? It's completely different. All the exposure is outdoors, even though it's the same similar mosquitoes, the same populations of mosquitoes that are primarily biting outdoors. In site 1, most of the exposure is indoors. And at site 2, most of the exposure is outdoors. And that's only because of the humans and how they are functioning and what their behaviors are. So that we found really, really interesting. And it was the first time we actually saw a big impact, because we measured it, a big impact of human behavior on exposure to mosquitoes, exposure to malaria rates. So where are bites happening? So again, if we take these figures and add up the number of hours people spend in each of these categories, we can actually get a pie chart. And you can see the red at site 1 is on the left, site 2 is on the right. So you can see the exposure outdoors, which is in green. At site 1, it's about 20%. And at site 2, it's, I don't know, it's a lot. At site 2, it was most of the exposure. The blue is indoors awake. And you can see at site 1, it's more than site 2 because people were outdoors more. And the red is people asleep without bed nets. And it's slightly under 50% at site 1 and about 25% at site 2. And you can see how people were outdoors a lot more at site 2. And the reason for it was really interesting. Remember, they went to sleep late and they were outdoors more. And the reason was because they had electricity. And that's just a simple explanation for changing human behavior, not mosquito behavior. The electricity changed human behavior. Because they had electricity, they stayed awake longer. It was light outside. They could play, they could cook, they could hang out, they could talk outside the house and get bitten by mosquitoes. And so the presence of electricity actually changed the exposure profile of that village. So now we went to the Ministry of Health. This was the end of our study. And we told them, here is where and when people are getting exposed to mosquito bites. This is how effective bed nets might be. This is how effective bed nets might not be. But here are possible intervention strategies you can implement, right? It's the same figure. When people are outdoors, you can possibly try LSM, which is larval source management or larviciding to kill the mosquitoes. You can possibly try topical repellents or spatial repellents at both sides. When people are indoors awake, since you cannot give a person a bed net and say, please walk around with a bed net around you, right? They're awake. So maybe spatial repellents or topical repellents would work. And in the red part, when people are asleep without bed nets, maybe you could increase bed net usage. So for the first time in Panama's history, they decided to do an experiment. Can we distribute bed nets to the single village, to everyone in the village, and see what happens to exposure? So that's what they did. Only in site 1, they tried to see if they could distribute bed nets, and with SBCC, which is social behavior change communication. So we went to the villages, gave everyone a bed net and told them, here is where our data says you are getting exposed to mosquito bites. Here is where your malaria is coming from. maybe if you use bed nets, you can protect yourself more. And let me show you what happened. So this is the same figure as the one at site 1, the same figure. So this is the figure on the left from March 21/9. It's the same one. So they did a bed net distribution plus social behavior change communication, where we communicated with the village elders and the village in general and told them how they could protect themselves. And the color went bad. I'm sorry. This big red part should be green. I don't know why that happened. I'm sorry. You can see, so the green, the outdoor exposure went from about 20% to almost more than 75%. So I'm sorry, this big part on the right should be in green. The red part, which is the same, went down hugely by over 90% because people asleep not under bed nets. Now they were under bed nets, so they're not getting exposed. And the blue part, which is people inside awake, also went down a bit. So, and this happened from March 2019 to July, August 2019. That's how many months? March, April, May, June. 5 months. In the space of 4 and 5 months, we reduced the exposure that was happening when people were asleep without bed nets hugely. But the important thing is that, let me get to this figure. So this is proportions, right? This is the proportion of exposure. And here you can see, on the top left, you can see it's correct over here. It's more visual. The green went up proportionally because more people were getting exposed outdoors. The red went down hugely because they started to use bed nets when they went to sleep. And the blue went down a bit too, because with social behavior change communication, people could see, how can we protect ourselves? How can we protect our children? So they wanted to go under the bed nets. And now they had bed nets because the ministry distributed it. And so the exposure profile changed. So even though this is just proportions, when we look at actual numbers down here, you can see pre-bed net distribution plus SBCCC and post. Inside awake, people were inside awake a lot, but that went down because people wanted to go under their bed nets to get protected with the SBCC. When they were asleep without bed nets, that was a lot of people, and that went hugely down because people now had bed nets, and with SBCC, they wanted to use them. And the exposure outdoors, right, remained the same. So people were outdoors just as much for the same time periods, and they just stayed outdoors the same amount. So the exposure did not change, but proportionally, this outdoor exposure went up, as you can see in the pie chart. Like, I hope that made sense. I'm trying to demonstrate that some exposure remained the same, which is outdoor exposure, but other exposures indoors went down significantly. So people were protected a lot there in those spaces and times. So when we answer the program's question, what was the program's question? The program's question was, are LLIs, are bed nets an effective vector control tool? Their question was this because they wanted to know if they should be distributing bed nets, because they did not want to distribute it, because everyone said the mosquitoes bite outdoors. Why would you give them bed nets? That's why they never did that. And the answer is, based on our data, that bed nets are an appropriate intervention. And they will be effective. They will hugely reduce exposure inside houses as long as people use them. But they are not enough to eliminate malaria because we have continuing exposure when people are awake and when people are outdoors, right? And with a primarily outdoor biting vector, even if people are mainly exposed indoors and have a good intervention indoors, they will still be exposure outdoors. So bed nets are appropriate. They are working well, but they are not enough as a single intervention. Other interventions must be trialed. Other interventions may be used to address this gap in protection. And as you can see, we've actually measured and understood this gap in protection. The gaps of protection are, the 3 different ones are continuing numbers of people who did not use bed nets when they went to sleep, a smaller number of people, but that still happens. People when they're awake inside their houses and cannot use bed nets, and outdoor transmission. Again, this should be green and not dark red. So you can see the continued gaps in protection. And possibly we can trial these other interventions, spatial repellents, topical repellents, larval source management, to combat these areas and spaces of exposure. Right. So the key messages are, I'm almost done now at almost 1.5 hours. The key messages are entomological surveillance can actually be the intervention. It'll tell us what's happening at our sites from all these perspectives. If we do this well enough, we can understand gaps in protection. And this is super important because this is where all our malaria is coming from. This is where all our diseases are coming from, where we are not protecting our communities, and understanding how we are not protecting them and where this continued exposure is happening will enable us to figure out how to improve our intervention strategies. We will make informed decisions about appropriate interventions, and it'll also help programs manage expectations. So this is something that was actually we did not realize would be important. And the Ministry of Health in Panama actually told us, with this previous slide, right, you can see how malaria, this previous slide, you could see that, let's say, the exposure was about 50% indoors. We could actually reduce this exposure by 50% by having people reduce their bed nets. And that's exactly what happened. We reduced exposure by almost 50% by giving them bed nets with SPCC. So instead of going to a Ministry of Health and saying, you should give everyone bed nets, and the Ministry of Health thinks, oh, we're going to eliminate malaria. The more realistic thing to say is, yes, bed nets will be effective, but you can possibly combat only about 50% of malaria exposure. Right? And so you manage your program's expectations. They have a more realistic understanding of the impact of these interventions. Too many places think, oh, we're going to put in this new intervention and get rid of malaria, right? That's not true. It's a lot more difficult than just that. So in short, evidence-based decisions will reduce malaria if we use entomological surveillance to ask appropriate questions, if we can get this data, analyze it appropriately, and then respond with suitable intervention strategies, suitable vector intervention strategies, we can actually use entomological surveillance as the intervention, along with, of course, bed nets and other things like that that are coming up. So evidence-based decisions will reduce transmission. And just as a note, this is, I think, my second last slide or my last slide. I'll have a sip of water. In 2021, the last malaria report, World WHO malaria report, there are about 247 million cases of malaria. That is 28,196 malaria cases per hour. That's 470 malaria cases per minute. And I have been talking for, I think, 1.5 hours. So that's 60, 70, 80, 90 minutes. And I'm looking at my calculator over here now. So 90 minutes times 470 is equal to, so since I started talking, there were 42,300 cases of malaria. That is ridiculous. And with 619,000 deaths in 2021, which is 70 per hour, which is 1.2 cases into 690. So since I started talking, about 108 people died from malaria, which is not okay. And even though when we talk about things like this, they're numbers and cases, just numbers, right? All I mentioned are numbers here. And I just want to demonstrate how these are not numbers. These are actual people. These are actual lives. This is in Namibia, where I was waiting for the military. I was staying close to this health facility, and the Ministry of Health said, "We pick you up from the health facility, please." Yeah, they said, "Please go wait there, and we will come pick you up to go to the villages to conduct some studies that we were doing there in Namibia." And I was waiting there, and there was this woman. She was with her little baby, and she's just walking back and forth in front of the health facility. And this man, at the same time, he came, and you can see his bike here. So he actually rode in on the bike, and he left the bike here, And he went inside and he came out about half an hour later and he laid down over here. And I was just sitting there. I was just sitting there waiting.
Dormancy? वो है हमारे पास.
Yeah.
सब कुछ.
Yeah, I was just sitting there waiting for the vehicle to pick me up. And what I didn't realize is that this woman was actually carrying her baby who had died of malaria. She came in earlier on in the morning and her baby had died and she couldn't let go. She was still really upset and she was just walking back and forth, holding her dead baby, which was horrible. And then this man who had actually biked, I think it was 25 kilometers. He lived in a remote village and he was sick with malaria and he couldn't get anyone to take him to the clinic. So he had to get his little bicycle and he biked 25 kilometers, sick with malaria. And he went inside and I don't know if he got treated or what happened, but then he came outside and he lay over there because he was just so tired and he died there. Like while I was standing there waiting for them to pick me up, this woman's baby died and she was like suffering and really upset holding her baby. And this man drove up and just died in front of me. You know, just he was so tired and so sick, even though he had biked that distance. So I just want to say that these are not just numbers, these are actual people. And like it's our responsibility, you know, our responsibility is not just to do what we're told, not just to be solution implementers, that we have to sort of change our mindset to be problem solvers so we can try and figure out how to address this problem and protect people and and use surveillance, our national strategies as an intervention ourselves. So I think that's it for now. I hope that was useful. 1.5 hours, not bad. We have half an hour for questions. Please let me know. I'm happy to answer any question at any point about my work.
Thank you very much, Neil, for the outstanding comprehensive presentation. That I think this is really valuable for us, not only for the student, but also for the program, malaria program and vector-borne disease program that also attend in this afternoon. So I think it's really, really good for us. So I think we still have time for maybe discussion. So I would like to open the floor for the question or comment from the participants. So before that, maybe please give the applause, big applause, I think, for the Neil. That's very really, really incredible presentation.
Thank you.
Okay. Okay, maybe any participant that have a question in this floor. Okay, maybe from the chat room, I see a question from the Khairil Anwar. So, may I ask Prof. Neil about the vector-borne disease protection, especially regarding the gap protection with other vector-borne disease like dengue. And then the implementation Bagaimana has successfully reduced the incidence of the dengue fever. However, the larva density remain high in the area, and there have been no disease transmission. So how do we view the gap of protection in the context? Yeah. So please, Neil. Okay.
Yeah, I hope I can answer the question appropriately. So the first question with gaps in protection with other diseases such as dengue, it's the same concept, right? So with Aedes mosquitoes, they have different behaviors. They don't bite at night like Anopheles. They bite more in the evenings and mornings and throughout the day as well. Actually, I'm being bitten by Aedes right now over here. So their behaviors are different. Their movement is different. So understanding their behaviors, like they don't travel very far. Anopheles can fly 2 kilometers maybe in a day, but Aedes do not. So understanding their behaviors and how the interventions work with them will tell us about the gaps in protection. So with Aedes, a lot of the times it is based on their larval sites and the presence of larval sites. And since they bite at times when people are usually not under their bed nets, like right now, me sitting here and being bitten by Aedes right now, that's a gap in protection, right? So I could possibly use a space repellent or something like that. So each disease, whether it's sandflies like kala-azar, or dengue, chikungunya, look at the vectors, look at the interventions that are present, and you can sort of try and understand their gaps in protection. With regards to Wolbachia, you're right. So you can actually take out the parasite, take out the disease, and you'll still have humans and you'll still have mosquitoes, but you won't have the disease. Like in the US, malaria was endemic in the US, and now there's no malaria there, but there's lots of vectors, mosquito vectors of malaria in the US. So with Wolbachia, you can have lots of larvae, but no disease, because you've eradicated the disease in that area. Not eradicated, eliminated that disease in the area. So that is a possibility. Gaps in protection with regards to Wolbachia, maybe I didn't understand the question appropriately, but yeah, if you look at the disease and the interventions present and the vector, you should be able to piece together what the gaps in protection are, where people are continually getting bitten by infected mosquitoes. I hope that answered okay. Yeah.
Okay, maybe any others question from the participant in the floor? Maybe you can raise your hand, maybe. Any other question? This is really a good opportunity for us to discuss with one of the most influenced expert, I think. Okay. Any other question?
Maybe I can ask, what kind of problems are you all seeing, any one of you, with regards to vectors or your diseases that you all are working on? What kind of issues are you all having at your sites? Are there any thoughts about that? What kind of issues do you all have? If there are no questions, feel free to interrupt me and ask a question.
Please, from the floor, maybe. Many people from many colleagues from Pakistan is also attend here. I think if you have also some answer from the nail question. Oh, so this is
yeah, that is correct.
Resistant. Yep.
So insecticide resistance is one of the things that they evolutionarily pushed back, right? So switching insecticides based on the intervention being used. So there are new different kinds of bed nets now that have multiple insecticides in them, like chlorophenapir and things like that. So if your mosquitoes are resistant to pyrethroids, which are the usual insecticides, you can switch your mosquito nets to bed nets with other insecticides or synergists like PBO, so you combat the resistance. So that's a good point from Susanna, I think. Yeah.
Yes. And many, maybe this is once more a question from Ken's, Neil.
Yeah.
Chemotherapy against malaria has shown to be a promising intervention as shared from WHO. Do you have any experience on implementing this strategy? And what are your interesting observation on this intervention? Yeah.
So what do you mean by chemotherapy? Do you mean just treatment with artemisinin and chloroquine and all those? If you mean that, you're absolutely right. That is, a treatment for malaria is very useful and very good, and there are multiple kinds of it. And yes, I am involved in some of it. And some of the ways of doing it are, for example, SMC, which is seasonal malaria prophylaxis, where you treat people before they have the disease. For example, in the Sahel in Africa, there's during the rainy season, malaria goes up. And the high risk groups are kids under the age of 15. So even before malaria starts going up, you start treating everyone. So seasonal malaria prophylaxis, you treat everyone and you can get malaria rates down in those children. You can also do mass drug administration. So treat everyone in the area before they get malaria, or everyone at the time. Or you can test and treat them, which is another way. So instead of treating everyone, test everyone, and whoever's positive you treat. But then, of course, there is just the normal health facility-based treatment, diagnosis and treatment. There's also prophylaxis. So, for example, this is just mainly for I don't think it can be done on a population level because it will be very expensive. For example, if I live in the US and if I go to Papua and there's lots of malaria there, they will give me Malarone or Mefloquine. And so I will take it just for the period of my travel so that I won't get infected. And of course, that's because my immunity is so low living in the US that if I do get sick, I will get very sick with malaria. So it's to protect me. So prophylaxis is an intervention mainly used for tourists, but it's not necessarily one that can be scaled up to an entire population because it's just so expensive. There is also resistance to a bunch of drugs out there, like artemisinin-based resistance. Artemisinin is a primary drug. And there is a resistance popping up in Asia, as you know, and now across Africa as well. And another problem that is happening is the HRP2 and 3 mutants. So the parasite, when we diagnose this is so interesting actually to me. So imagine if you have parasites, right? People are infected with malaria. And then you have RDTs, which are rapid diagnostic tests. So you take a drop of blood, it's like those pregnancy tests. You take a drop of blood, you put it on the test, and it detects a protein, a parasite protein, like a pregnancy protein. And if you have that parasite is there, you have the protein in there, and the line will show up. And you'll be like, yep, you have malaria, or yes, you're positive, right? You're pregnant. It's like a pregnancy test. So if you imagine, so every single parasite that gets positive will get treated and you kill those parasites. So now there are some parasite mutants who don't have functioning, or that little region that you detect of that protein is gone. It's a deleted. So even if you're positive with malaria, if you have malaria, you come out negative in the test because you cannot find that protein. So you can see how the parasites are also evolving to combat. And so those people don't get treated because they don't show up as being positive at health facilities. So that's becoming a problem as well. Yeah. I hope I answered your question, Ken.
Yeah, thank you. Maybe Neil, maybe any other question from the floor? If not, I just maybe asking about the role of the species complex, Neil, in related with the malaria transmission, because of we have in Indonesia we have a problem related with the very difficult elimination in several part with the occurring the presence of the species complex. What do you think about this, Neil? Maybe you have a suggestion or maybe some the previous study that spotted this.
Yeah, so you are absolutely right, Paktri. So species complex are actually really confusing to work with. And a simple way of describing them is that mosquito species, we identify them by how they look different, right? They might have a different banding pattern, or they might have a different line on a wing or on a leg. And each of different species look slightly different. So we can look at them and say like, oh, this is species A, this is species B, this is species C. But sometimes you have a species complex in which they're actually different species, but they look exactly the same, and you can't tell the difference. And the problem is that each member of that species complex might have different behaviors. And some of them might be vectors, and some of them might not be vectors. An easy example is in Africa, there's the Anopheles gambii complex, and it has up to 7 members of the species complex. And depending on the site, some of them don't transmit malaria, so you can ignore them for malaria. Some of them will feed indoors. One species will feed outdoors. So if you look at them, you think it's, oh, Anopheles gambii. But depending on the species, you might have a completely different exposure profile. In some sites, it'll look exactly the same, so you think it's one species, but it's actually all the transmissions outdoors, and actually all the transmissions indoors here. So understanding species complex can be very important. In Papua, for example, where the transmission is very high, there are multiple, each species actually might have, there are 3 main species there, Anopheles ferrati, Anopheles punctulatus, and Anopheles coliensis. We might have species complexes, and we actually do know we have species complexes present, and some are vectors, some are not, different behaviors. And we don't really understand this, and understanding it really well. If you imagine the little lecture I gave, the same sort of way, if we understand which site has which species, members of which species complex, and what they are and what their behaviors are, it might be really helpful in targeting and tailoring our intervention strategies to them. Yeah, did that make sense, Bakri?
Yes, of course. Thank you very much, Neil. I think it's really, really important to understand the species complex, right? Yeah.
Right. Absolutely. Yeah.
Okay. Thank you very much, Neil. And maybe, is there any question, or maybe enough for us? Okay, if there is no question, so thank you very much, especially for Professor Lobo, that's already present very, very interesting, and I think it's really, really incredible. And thank you very much for your time, because I know that you are very busy. So this is a really, really yeah, opportunity, big opportunity for us to learn from you, especially. Yeah. So now, thank you very much for all of the participants, especially the active participant, and then so we appreciate your engagement and contribution to the discussion. So we hope you found it informative and valuable information from this lecture. So I think all of my class is already finished. So I will hand over the session to the talented master of ceremony. So thank you very much, Neil. So I will send back the my turn to the MC. Thank you very much.
Thank you, Padre Wibowo.
Yep.
So that was it, ladies and gentlemen, the magnificent lecture from Prof. Lobo. So I would like to appreciate today's webinar as well to the participants. As Padre Wibowo already mentioned, we can see from the chat box that there are some entomologists from Pakistan joining us. Thank you. So to capture this beautiful moment, we'd like to invite all of you to open your video camera so we can have this virtual photo session together. All right, so is everyone here? So there will be 4 parts. Yeah, there will be 4 sessions for this virtual photo session. The first one, here we go on my count in 3, 2, 1, cheers. Thank you. Let's move on to the second page. We have, okay, in 3, 2, 1, cheers. To the third page. In 3, 2, 1, cheers. Last but not least, to the last page in 3, 2, 1, cheers. That was it, ladies and gentlemen. Again, thank you so much. I still remember that Prof. Lobo suggested us to use the mosquito repellent. He also mentioned Autan, but he is not the brand ambassador for the brand. Definitely, right? And atas nama panitia yang bertugas, my name is Rima. Thank you so much. Please update our information regarding the webinar lectures on our social media at Tropmed UGM, and kindly fill in the attendance form on the chat box, because your e-certificate will be delivered based on the data that you sent to us. Thank you. My name is Rima. I'm signing out. Wassalamualaikum warahmatullahi wabarakatuh. See you. Bye. Thank you.
See you.
Bye.
Thank you.
You.
Bye everyone. Thank you.
Bye.
Yeah.
Bye.
Yeah. Ready.
Makasih Pak Tribo, matur nuwun sangat. Sampai ketemu.
Oke,
Sama-sama,
matur
Ibu.
nuwun.